All right, given that I am recording, I'm gonna just start. Hope people join in as we talk. Hello everyone, welcome back to our computational genetics discussion group. We return now in 2021 with a great speaker. Gregor just snatched for me. Please feel welcome, Vince. Before I pass on the mic for you, just a few reminders that, well, please keep your videos and microphones off if possible. And if you have any questions during this presentation, you can always use the chat box and you can ask those questions throughout the presentation. If I see fit, I'm gonna interrupt Vince, if that's okay, Vince, for you. So we can have a nice discussion through all this meeting. And yes, and I'll be recording from every session from now on. So yes, please, if you can authorize the recording of this meeting. I think Zoom pops in a chat box, but anyways. Let me just introduce our speaker. Vince is from the University of Oregon, and I believe he's going to be talking everything about linked selection for us today. And I'll now pass on the word to you, Vince. Enjoy.
Great. Thank you so much for inviting me to present some of my work. So the sort of theme, as you were saying, is link selection, and the title is Understanding the Effects of Link Selection on Two Timescales. And this is some work that I've done. There we go. This is some work I've done towards the latter half of my PhD with Graham Coop at UC Davis, as well as some work that I've done during the first year of my postdoc here at the University of Oregon. So the first part of the talk is sort of looking at short-term effects of link selection. So thinking about things over the scale of tens of generations. In particular, I'm going to talk about this idea of temporal covariance, which is sort of a signal to detect the short-term effect of link selection from polygenic selection. And then I'm going to talk about how we can use temporal covariance to estimate the genome-wide contribution of selection to allele frequency change, and apply this to two different evolve and resequence studies. And then the second half of the talk, I'm going to sort of explore Lewinton's paradox, this long-standing mystery in evolutionary genetics, and tackle that from a few different angles. And then finally, at the very end of that, see if link selection can explain this paradox. Okay, so we have countless examples of traits evolving over very short timescales. You can go out in nature and see things like Darwin's finch's beak shape changing over the scale of a couple decades. There's also this nice experiment done by Steve Frank, where he looked at flowering time before and after a drought and saw a really strong shift. And you might imagine that given that we can see traits changing on these rapid timescales, which is something that artificials or breeders have known for a long time with artificial selection, we could imagine that if we were able to sequence these populations through time, we could somehow look at the allele frequencies changing and then suddenly say, oh, this is consistent with adaptation. We see this at the genomic level. However, many of these traits that are being selected on these timescales are polygenic, meaning that there's hundreds, potentially thousands of variants determining the trait's value. So what's difficult about this is that at any one particular spot in the genome, the sort of effect of selection is dispersed across everything. So at any one particular spot of the genome, the allele frequency changes are very, very subtle and likely indistinguishable from random genetic drift and sampling noise. So what I'm going to try to motivate here is that we want a way to quantify genome-wide allele frequency changes and sort of assess whether these are driven by selection, whether they're driven by heritable fitness variation differences between individuals. So there's a lot of approaches out there in the population genomics literature that sort of take temporal data, and they have the goal of explicitly finding selected loci. And they're sort of using this signal that under selection, we would expect that allele frequencies would change faster and maybe more in one direction than under random genetic drift alone. By contrast, the work that I'm going to present here is a little bit different. So our goal is to really just take this cumulative set of allele frequency changes through time and really try to develop sort of a signal to detect whether genome-wide there's evidence of selection occurring and in particular on a polygenic trait, which is rather difficult to detect. And the signal that we're going to be using in particular is this idea of covariance in neutral allele frequency changes, which is something that only linked selection can create. So just to sort of give a quick introduction to the temporal covariance idea, we can imagine looking at an allele frequency change through time, here from some initial generation 0 to some later generation t. And we can think about this allele frequency changing and then breaking up that total change from generation 0 to generation t into these delta terms, delta p0, delta p1, and so forth, which are just the differences between adjacent time point allele frequencies. So then we can write the variation in total allele frequency change as the variance in the sum of these delta p terms across all of these different intervals. And under neutral genetic drift alone, this variance of the sum can be rewritten as the sum of the variance. And what allows us to do this is that these allele frequency changes are independent of one another. And this is in part because under genetic drift acting alone, there's no selection. There's nothing that can lead to sort of correlated changes through time. However, under the case of selection, when there are heritable fitness variation differences between individuals, there are now covariance terms that contribute to this total variation in allele frequency change. And that contributes to this sort of total variation on the left side here. So this is actually something that connects with work that Alan Robertson did in the 1960s. It's sort of implicit in his results, and these ideas have been developed by many folks since then, including Santiago and Caballero, a series of papers in the 1990s. So I'm sort of expanding on this idea and seeing if we can detect this in temporal genomic data. So what's sort of the intuition for what creates temporal covariance? Well, you can imagine some neutral allele, which I've colored here in orange, on some beneficial fitness background, which is blue. And at some early sort of time point, we have some increasing allele frequency because it's on this beneficial background, delta pi. And then as long as this neutral allele remains sort of associated to this fitness background, it isn't completely recombined off this background. At some later time interval j to j plus 1, we expect this allele frequency change, delta pj, to also be sort of increasing at that point. And so if we were to look at this, this one is increasing, and then this one is increasing. This leads to a positive covariation in allele frequency change. By contrast, under neutral genetic drift alone, these covariances are expected to be 0. And I want to emphasize that had this neutral allele found itself on a deleterious background, it would decrease at some, between some generations here and then decrease at some later generations. This would be a negative allele frequency change and another negative allele frequency change, which again just creates positive temporal covariance. So, pardon me, regardless of what sort of background it finds itself on, this sort of effect is leading to covariance and allele frequency change. So in a, what year was this? 2019 genetics paper, Graham and I sort of developed the theory of temporal covariance. So if you're interested in a lot of the mathematical details of what drives this, just check out this paper. I won't be going into too much depth, but I will say that just roughly what is creating temporal covariance, how can we explain the magnitude of this effect? The two major components are the level of recombination, which are varied in different columns here. This is the level of recombination in a region in Morgan's. And then across different rows here, I have the level of fitness variation. And what I've done here is these are simulations for different numbers of loci that determine the trait that's being selected. And there's no selection occurring the first 5 generations. And you can see that the temporal covariances, which are on the y-axis here, are 0. And then once we have selection on the trait, the temporal covariances shoot up, and then they sort of decay through time. So why are they decaying through time? There's 2 major reasons why. One, the neutral alleles that we're sort of tracking are disassociating from their backgrounds due to recombination and segregation. And 2, the level of fitness variation is decreasing through time just because selection is removing this fitness variation. So in our genetics paper, we also propose a very simple measure of the impact of link selection that we can go out and sort of apply to data sets. And the idea behind this measure is that if I have a series of allele frequency changes through time, I can calculate these delta P terms, and then I can just calculate sort of the empirical variance covariance matrix of all these delta P terms. And then we can think about writing the variation in total allele frequency change, averaging over all the loci in the genome, as the sort of the sum of the variances, which in terms of the variance covariance matrix is just the sum of the diagonal elements. And in the case where neutral drift is acting alone, we would expect this to be the only thing contributing to this total variance. By contrast, if there's link selection, now we have the upper and lower triangles that are contributing to this total variance as well. And this suggests a very simple estimator of the impact of link selection, which is just essentially the sum of all of the covariance elements contributing to this total variance divided by the total variance itself. So again, under genetic drift acting alone, we would expect this G measure to be 0, whereas under selection, this would be greater than 0. Okay, so now I'm going to talk about how we've taken these ideas and applied them to some evolve and resequence studies. In the original paper, which is now, it was published last year in PNAS, we looked at 4 different studies. I'm only going to talk about these 2, Bargy et al. 2019 and Castro et al. 2019. So the first study, Bargy et al., is an evolve and resequence study done in Drosophila simulans. This is a really nice paper. What they've done is they've evolved Drosophila simulans for 60 generations in a novel sort of warm lab environment. And they have 10 independent replicate populations. And they sequence every 10 generations. So there's 7 time points total, including the initial generation, which allows us to sort of do lots of cool things with this data set. So the first thing I did is I sort of, as I was just explaining, I can calculate all these allele frequency changes across the genome. And then I can calculate the empirical variance covariance matrix, which really just quickly, it's a very simple statistic, but it can tell us a lot about evolutionary processes. So I can look at the rows of this variance covariance matrix, which are essentially the covariance between some initial allele frequency change. I'm going to show you this first row here. So this is the allele frequency change between 0 and 10, right? And then it's the covariance between that allele frequency change and then some later time point, which I'm varying on the x-axis here. And those later time points, here's this. Can you see my cursor, by the way? Is it, no?
Yes, we can.
Okay, good. Just checking. I'm like, yeah, okay. So this would be the covariance delta PS between where S is this change between 0 and 10, and then delta PT, which is this change as it varies along the columns of this temporal variance covariance matrix. So what we see is that consistent with what we'd expect under selection, these covariances are positive early on, and then they decay through time. So then looking at later time points, which are corresponding to rows of this variance covariance matrix, we again see that they're positive and they decay through time across the board, which is quite interesting to see. So I want to emphasize too that this is what we would expect, right, from our theory paper. We would expect that because of the decay in fitness variation as selection proceeds, as well as the decay in linkage disequilibrium due to recombination and segregation, that these temporal covariances would decay as we observe. And I've also done some simulations using the SLIM framework for this paper. And these are sort of more realistic simulations where we have a Gaussian fitness function. So we sort of burn in with stabilizing selection and then have optima shifts of different intensities. And we can see that basically the strength of the optima shift dictates the magnitude of temporal covariance that we see. So next, I'm going to sort of discuss estimating G, which is, again, this fraction of frequency change due to link selection. And what I'm going to be doing here is considering different subsets of this variance covariance matrix. So looking at the smallest subset that we can calculate this G measure for, which is just a single covariance and then 2 variances. This gives us a sense of basically how this quantity changes through time. We see that this estimate of G through time increases as you would expect under selection. And as we proceed and take larger sort of chunks of this variance and covariance matrix, calculating this ratio of the sum of the upper and lower triangles to the total sum, the fraction of variation in allele frequency change driven by link selection is increasing. and ultimately plateaus to at least 20%. And this is, I mean, I was pretty excited about this. I'll mention why it's at least in a second, but this is a pretty sizable portion of allele frequency change to be caused by fitness variation, especially in populations that are quite small, right? These are lab populations where there's on order of about 1,000 Drosophila per cage here. And this is also, as I mentioned, an incredibly conservative estimate. So why is that? Well, from the theory section earlier, I mentioned that the covariances are strongest early in time between adjacent generations. Here, because of the sequencing design, we're only looking at 10 generation blocks because these populations are sequenced every 10 generations. So we're actually missing, we can't directly compute the strongest covariances and put those into this G measure, which are between adjacent generations. Instead, we're looking at sort of these covariances between blocks of generations, which are pushing those other terms basically into the denominator and not the numerator of this quantity. Second, we know too that link selection is also increasing the variation in allele frequency change, but we don't have a way to directly measure that. So it's another reason why this is conservative. Okay, so next I'm going to talk about this artificial selection experiment in mice. So this is a really lovely paper where they have bred mice for larger tibia length using two replicate lines, Longshanks line 1 and Longshanks line 2. And then there's a control line where the individuals that are chosen to reproduce are done so with a random number generator. And over the course of some 20 generations of artificial selection, they've seen in these Longshanks lines a 5 standard deviation shift in tibia length relative to body size. Here, though, however, we only have 2 sequence time points. And given that the temporal covariances require at least 3 time points, this is a limiting factor to apply our temporal covariance theory. So instead, what I developed was this idea of a convergence correlation, which is based on this convergence covariance, which is very, very similar to the temporal covariance. So the idea behind this is that if we have a founding population, which 2 sort of independent replicate evolution lines are drawn from, and then sequence at 2 time points, we can compute the allele frequency changes across these 2 replicate lines, ending up with sort of in replicate population A, these delta P A terms, and likewise for the replicate population B. In the case where there's shared selection pressures, and there's sort of haplotypes that are common between these 2 replicate lines that are sort of drawn from the same founding population, we would expect, for example, if you were to select for tibia length, that if you have some tibia length increasing alleles on a shared background, that across these 2 replicate lines, both of these haplotypes would increase in frequency, and this sort of parallel shift in haplotype frequency and allele frequency would lead to delta P A terms here that are both positive, delta P A, pardon me, delta P B terms, and that leads to a covariance that's positive, much like temporal covariance. By contrast, if there's no shared selection pressure, or things are long since sort of evolved neutrally since they've been taken from the founding population, under drift alone, you would expect these allele frequency changes to sort of be random, there to be no signal of covariance across these replicate populations. So what do we see? Well, the design of this study allows me to compute basically 3 pairs of these convergence correlations. The first of which is between the control line and Longshanks line 1. Again, we would expect there to be no shared selection pressure between the selection line and this control line. And we see that the convergence correlation is indeed not statistically significantly different from 0. And likewise with the control line and Longshanks line 2. And then later in this Longshanks line 1 to Longshanks line 2 comparison, we see that it is statistically significantly different from 0, consistent with us detecting a signal of sort of parallel or convergent selection. So when we first presented these results to these authors, they were excited by this, but they suggested, though, that this could be perhaps driven by some large effect loci that they found in their original study, rather than sort of the signal that we're hoping to detect, which is polygenic selection. So to look at this, what I've done is I've removed not just the sort of loci that they've found, but actually the entire chromosomes that these loci were on from my analysis. And the idea here is I don't really know the extent to which that linkage disequilibrium in these particular populations, where it could be quite long, is extending over large stretches of these chromosomes. So the most conservative thing is just to drop the entire chromosomes entirely. And doing that, we find that our signal really isn't changed that much, that we actually, we think that we are detecting this signal over short periods of time of polygenic selection. So in my paper with Graham in 2019, we also hypothesized that if there's a reversal in the direction of selection, that these temporal covariances could become negative. And the idea here is that, if an allele frequency is increasing in time, that suddenly it changes and drops in frequency. And that leads to basically a consistent difference in the direction of selection and allele frequency change, creating a negative temporal covariance. So these are some simulation results where there's initially no selection on the trait and then selection for positive trait values, and it switches, and you can see on the y-axis, temporal covariance becomes negative. So if you're looking very carefully at these figures, you may have noticed that there actually is some evidence of negative temporal covariances, these later time points. And based on the bootstrap-based confidence intervals that we've generated, that these look to be significant. So I wanted to explore this a bit more because, realistically, these temporal covariance estimators, they're incredibly noisy, right? They're averaging over the entire genome. We're trying to pick out this signal of very, very weak selection. And there's lots of things that are sort of being washed out. So you can end up with maybe like in one stretch of a chromosome, a reversal in the direction of selection that's very significant, but may, just because of the genome-wide averaging effect, be washed out. So to look at this, what I've done is calculated these temporal covariances in windows along the genome. These are, I think, megabase windows at time points, sort of 2 time points apart. So this is this off-diagonal element here, corresponding to k equals 2. And I've calculated these windowed covariances, and at any one window, the signal is sort of too noisy to sort of say, definitively, oh, this is changing in the direction of selection. But the distribution of windows across the genome is actually informative about evolutionary processes. So this is consistent with what I showed earlier, which is that compared to this sort of neutral null distribution, I won't go into the details of talking about how we generate this for now, but compared to this neutral null distribution, there's sort of a heavy right shoulder consistent with an excess of positive temporal covariances. However, at later time points, compared to this neutral null distribution, there's now a significant heavy left shoulder, consistent with across the genome reversals in the direction of selection. And I want to say too that this is, I found, pretty interesting. So in this study, there's no intended reversal in the direction of selection. These are just, Drosophila they've taken from a population, thrown in this, novel lab environment, and there was no planned change. So this is potentially indicative of some complex selection dynamics that weren't originally intended. So I don't have time to mention some other nice details of this paper. I did a tremendous amount of simulations, including looking at optima shifts and how G changes through time, as well as basically evolving 2 populations independently, and then pushing them under some shared selection pressures and convergent directions and divergent directions, and we can see our statistics sort of behave as we would expect. Finally, I didn't expect this to be the case, but it turns out that background selection can actually lead to temporal covariance as well, which is in this top figure here. So these are all details that are in the paper. So if you're interested, you can check those out. Okay, so for the second half of this talk, I'm going to talk about this paper that was pre-printed last week, which is looking at Lewinton's paradox of variation. So what is Lewinton's paradox? So sort of in his synthesis of the find 'em and grind 'em era of population genetics, Richard Lewinton in the mid-1970s argued that there's something strange with neutral theory, because if you look at the range of heterozygosities across species as different as humans and Drosophila and horseshoe crabs, this would suggest that these are all within a factor of 4 of each other in terms of population size. And the figure in his book, which I really like, is this one here. And so this is the range of heterozygosities that he observed. This is the relationship we would expect under neutral theory, this nice curve here. And then looking at the changes on the y-axis, it would suggest that this product here between population size and mutation rate falls in this narrow range. And mutation rates do vary, of course, but not over as many orders of magnitude that we expect census population sizes to vary. This is the real mystery, right? Is that I think now we all know that what drives this isn't census population size, it's effective population size. But the mystery is that we know that census population sizes for things like lynx versus Drosophila simulans vary over many orders of magnitude. What is essentially decoupling census sizes from effective population sizes, which is a restatement of Lewinton's paradox. So there's sort of two views going back to the 1970s, 80s to present day on what could lead to this decoupling. The first view is that neutral processes do this. So Kimura was arguing this in his book, such as recurrent bottlenecks, especially since the last glaciation. And essentially mutation drift equilibrium takes a while to reach, and it takes longer to reach the larger the population is. So this is one thing that can decouple census sizes from effective population sizes. Another thing is variance in reproductive success. So organisms like this Pacific oyster here on the bottom right have incredibly large variance in reproductive success, and that also acts to sort of decrease effective population size. There's also work done that sort of argues that colonization and expansion dynamics can also change things quite considerably. So the other view is that selective processes could drive this difference, right? That this could lead to effective population sizes being reduced from what we would expect from their census sizes. And actually, Lewinton's paradox was the impetus from the Maynard Smith and Hague hitchhiking model. And we now know from a variety of theoretic work and empirical work that link selection comes in many different modes, recurrent hitchhiking, background selection. There are these quantitative genetic models of link selection, which are like Robertson's, which I talked about in the first part of this talk. And we have, across many different species, evidence that link selection is drastically reducing diversity across genomes and within species. So the questions, what I'm sort of revisiting Lewinton's paradox in this paper that I hope to address is, what is the relationship between genomic estimates of pi? So this earlier work was all sort of allozyme-based estimates of heterozygosity. Here I'm looking at genomic estimates of pi. So what does this relationship between genomic estimates of pi and census population size look like? And in particular, how big is the shortfall between observed pi and the predicted level of pi were we to do the things sort of that Lewinton suggested, which is to say that the census population size is what's driving things. Second, is this relationship between pi and population size significant considering phylogenetic non-independence? And then finally, can link selection explain Lewinton's paradox? So there's a lot of early work done looking at this relationship between sort of population size and heterozygosity. And generally, it's been found that it's a poor fit to what we would expect if census size were equal to effective population size. And I wanted to sort of increase the number of taxa and use genomic pi estimates to get a newer picture of this. However, the difficulty is estimating census population sizes for a large number of species. So the approach that I take is crude and approximate, but I think for these purposes, it's completely sufficient. It's basically saying that the census population size we can think of as the range, which is in kilometers squared, and the density, which is in individuals per kilometer squared. And then what I'm going to do is estimate the range from occurrence data. So there's this GBIF resource, which is fantastic, and you can go through and download occurrence data that's GPS tagged for many species. So I developed sort of a computational pipeline that does this, and I end up with a picture like this. So the data points are for this is a type of B. The data points are these red points here, which are the GPS tagged occurrence data. And then I sort of have a geometric approach that takes this and estimates the range from this, which is shown here in green. So this is honeybee, Apis mellifera, which is much more cosmopolitan. You can see that the estimated range is much larger. This is wolf here, and again, really large cosmopolitan distribution. So I'm doing this for 172 taxa, and I end up with a set of ranges which are sort of broadly consistent with earlier sort of discrete or categorical estimates. This is from Ellen Leffler's study in 2012. And you can see that things like that she categorized as island or narrow endemic have smaller estimated ranges than cosmopolitan species using my measures. So the next part of this estimator is population density. And here what I'm doing is using some work from the macroecological literature, which uses body mass basically to to predict population density. And John Dameth in the 1980s showed in a few papers that there's this surprisingly strong relationship across deeply diverged metazoan taxa between population density and body mass on this log-log scale. So, species as different as aquatic invertebrates and mammals you can see they have this very tight relationship that's homoscedastic and allows me to basically take the body mass of a species and then predict its population density. So that's what I'm doing here. For the sake of time, I won't go into too many details about this, but I basically measure body length, since that's a lot more standardized across species. I use another sort of relationship here on the bottom right to predict body mass from body length. And then from body mass, predict population density, given this Dameth relationship, and then use my estimated ranges, take the product to estimate population size. And this is all done in a scan routine, this Bayesian computational framework, which makes this kind of stuff quite easy. So what do diversity estimates look like against population size estimates? So this is this figure that sort of shows, I think, an updated version of that Ney and Grau picture and the Soule picture, which is on the x-axis is approximate population size on a log scale. And then on the y-axis is pairwise diversity. And you can see that across species, there is this increasing relationship between diversity and census population size. I've also shown here for reference sort of what we would expect if the effective population size were equal to, pardon me, the approximate population size on the x-axis for a range of mutation rates between 10 to the minus 8 and 10 to the minus 9. And you can see that overall the relationship is really different. It doesn't increase here. Let's see. So there's an order of magnitude shortfall for these large census size species between predicted diversity levels and observed diversity levels. And then there's no increase in this region. And this is sort of what Lewinton pointed out was this problem is that, in this sensitive region, as he called it, you would expect an increase in diversity, but the signal is pretty flat. So next, I wanted to consider the problem of phylogenetic non-independence. And this is something I've been curious about for a while. So in the 1990s, John Gillespie in his book looked at actually the Ney and Grau data, and he said that he sort of conjectured that this is likely this relationship between population size and heterozygosity is spurious because these large census-sized species are predominantly Drosophila, and these low heterozygosity, small population size species are predominantly carnivores. And if you sort of remove these two clade level effects, this relationship is a lot less clear. And this is fundamentally, akin to Felsenstein's 1985 problem. And surprisingly, since then, no one has really, to my knowledge, been using phylogenetic comparative methods in population genetics, even though there's a lot of across species population genetics happening. So I wanted to try to assess my pairwise diversity population size relationship in the light of phylogeny. And so what I did is I used a synthetic calibrated phylogeny for nearly all of these species, which is made possible from this great software package called DateLife, which allows you just to sort of develop a calibrated synthetic phylogeny for an arbitrary group of taxa. And what I find is that the relationship accounting for phylogeny is somewhat attenuated compared to just the linear regression line. So the linear regression line is this gray dash line here. And then the phylogenetic mixed effect model estimate is this blue dash line here. So the relationship is still significant, but somewhat attenuated. And perhaps more surprisingly, and more importantly, is that the degree of phylogenetic signal in this data is immense, right? So this is a very high degree of phylogenetic signal. So in some sense, Gillespie's concerns were correct. I mean, the relationship is still significant, but it's driven by really strong, deep splits between chordates and non-chordates in these data sets, corresponding to sort of our intuition that chordates typically have, lower diversity and smaller population sizes than invertebrates. So finally, in the last few minutes, I just want to sort of look at this question of whether link selection can explain Lewinton's paradox. So there's this idea, we can sort of simply represent this idea of the impact of link selection as there's some initial level of diversity in the absence of the effect of selection, which is called pi0 here. And then there's some reduction factor. And then there's some observed diversity level, which we can go out and see. And the difficulty here is that we can't directly observe pi0. And there's lots of really nice work done on using models of link selection to try to estimate this effect. And so we can't observe pi0, and we also don't really have estimates of the reduction factor for most species. So there's work done in humans and Drosophila and mice that estimates the impact of link selection, which can sort of give us these estimates of R across species. But for those 172 metazoan taxa, there's just not currently the genomic data, the population genomic data to estimate these things. So the question that I'm getting at here is a bit different. I was thinking that like if I don't have this data, what can I look at with this relationship between pi and diversity? Well, I can ask an interesting side question, which is if background and selection and recurrent hitchhiking were as strong in all of these species as they are in Drosophila melanogaster, where we have very good estimates, and we know from a lot of work that Drosophila melanogaster has incredibly strong link selection, and its pairwise diversity is strongly impacted by these types of selection. Could link selection solve Lewinton's paradox if pi0 is basically driven by the census size here? So I'm going to be using these Drosophila melanogaster estimates of sweep and background selection parameters. And then another factor that mediates the impact of link selection that varies across species is the recombination map length. And this is something that I looked at using this nice data set from Jessica Stapley, and where she's collected map lengths for across a broad variety of species. And I find that there's actually this surprisingly strong relationship between recombination map length in Morgan's and my estimate of approximate population size, which would suggest that species with really large population sizes have much shorter map lengths and thus would experience greater effects of link selection. And whereas, sort of smaller population size species have much longer map lengths and everything is much more loosely linked and things are less affected by link selection. So I consider the impact of recombination as it varies across species using this data set. And what I find is that if we were to use sort of Drosophila estimates of the strength of link selection, and this is to be more specific here, this is the number of sweeps per base pair and the probability that two lineages are trapped in a sweep. Those are the key sort of parameters that I've taken from Drosophila. So if we were to take these strong, sort of selection parameters from Drosophila, and we assume that the effective population size is equal to the census population size, I can predict under these, background selection and hitchhiking models what the reduction in diversity is, right? And essentially doing that for all the species, keeping in mind their own map lengths, and then for a range of mutation rates here, this is sort of the curve that we would expect, if everything were as strong as in Drosophila melanogaster. And what's interesting is that, at the high sort of census size species, this can reduce diversity a lot. However, in these sort of mid census size species here, their map lengths are essentially too long to really fit the data well. There's the effective link selection is much weaker here. And we could bring this curve down, but that would imply that there'd either have to be stronger rate of sweeps in these mid census size species and in Drosophila melanogaster, or much higher levels of background selection, which both seem unlikely, very improbable. This is really recapitulating a concern that Graham published in 2016, sort of arguing the same thing when he reanalyzed Russ Corbett-Dettig's data. So overall, this seems to suggest that link selection would have a really hard time explaining Lewinton's paradox. Finally, I just want to say too is, while under these simple models of link selection that I'm using, predicted levels of diversity can be reduced to observe levels for things in the Drosophila census size range. This would also basically imply that we're currently underestimating pi0, the absence, the pi and the absence of link selection by several orders of magnitude, which again also seems rather unlikely. So overall, I would say that my prior on whether link selection can explain Lewinton's paradox shifted a lot throughout this project. And I'm much more in it's probably some sort of demographic recurrent bottleneck type process. And in the discussion of the paper, I also talk a lot about the role of macroevolutionary processes as well, explaining this. So I've skipped a lot. You can check out the preprint, Why do species get a thin slice of pie? Revisiting Lewinton's paradox of variation. So with that, I'm happy to take questions. I'd like to thank these folks.
Thank you very much, Vince. I'd like to encourage you using the chat to ask questions, and we already have one from Gregor. So, yes, can you read the chat, or would you like me to?
I can, yeah. So it says, you presented a method to infer the amount of change in allele frequency due to link selection on a polygenic trait, fitness. In one data set, you showed that 20% of change is due to link selection. Does that mean the other 80% is due to direct selection on the loci? What type of data, whole genome, sequence versus SNP array affect results? Okay, so there seems to be two sort of questions here. So in terms of the direct effect of loci, so it's a good question. This was something that, yeah, a reviewer raised, is that, I'm sort of, in my theoretic work, I'm considering these being neutral alleles, but you may, we know that alleles are directly selected and we may sequence those alleles. So we can imagine, I mean, it actually, in terms of the mathematics, doesn't make that much of a difference. You can just imagine that if we were to be looking at a truly selected allele, mathematically, it's the exact same as a neutral allele that is, 0 base pairs, or like recombination fraction 0 away from a selected allele. So these things can be under directional selection, but the temporal covariance, sort of mathematics, it behaves the exact same. And then what type of data, whole genome versus SNP array, would affect results? So I haven't used a SNP array. I imagine, besides the usual issues of ascertainment bias, that, there could be some issues there, but I imagine it should work. I think, to my knowledge, no one has tried this on RAD data. I don't know how it would behave in that case, but I've applied it to other data sets since this original paper, and it seems to be working. So I imagine it should be fine. All right, we have another question here. I'm curious if you can say something about the relationship between the impact of link selection as measured by your covariance and the effects of a genomic level in terms of segment haplotype length. Oh, this is a good question. So let me think about this. So yeah, initially we had planned, and this is still some work that I want to do, we had planned to develop a sort of a composite likelihood-based approach to sort of say, like, here's this neutral allele, we observe some allele frequency change, and then much like Elyashiv et al., which is where I get these Drosophila selection parameters in the second half of the talk, we can, sort of imagine, that there's this neutral site, we're observing its allele frequency changes, it's this recombination fraction away from all of these other sites, and actually develop a model-based approach that considers, basically all of its neighbors and how those neighbors are perturbing its allele frequency trajectories through time. That's still in the works. Essentially, a lot of the machinery is developed, but unfortunately, the sort of data sets where we have temporal data for, they're not, they don't have linkage disequilibrium data, which is like a key parameter that's necessary. So I hope to, at some point, basically be able to use this information more for actually, I mean, ultimately to estimate added genetic fitness variation through time. So in our genetics paper, we show that with a very, very simple statistical technique, that we can estimate the additive genetic fitness variation through time from genomic data, which I think is really exciting synthesis of population genetics and quantitative genetics. All right, so let me see. Is there any where the method would break down and most change due to link selection? Asking because, oh yeah, I see. Sorry. Is there an effective population size where this method would break down and most change is due to link selection, 80% instead of 20%? Asking because intensely selected breeding populations, NE can be very low. So the census size for the Castro et al. Study in mice, that was something like 20. I think that was the NE. It's also a very, very strong sort of, there's a very, very small population size. So I think everything should work there. And this is a case where if you were to sequence every generation, I mean, what we could say is just so much greater. I mean, we could tease apart the effect of selection. And like I said, that estimate of 20% is really conservative, and most likely we would expect it to be much greater if you were to sequence every generation. So I hope that gets to your question. Question from Adam here. Hi, Vince, what is the slope of the relationship between log diversity and log census size with and without the phylogenetic correction? Okay, so this is a really good question. So the I can't remember actually. In the paper, I translate it to something meaningful. I think it's, let me think about this. It's 0.09 differences per base pair for every order of magnitude increase in census population size without the correction. I don't know off the top of my head what it is with the correction. But it is somewhat attenuated. So the regression lines sort of are a little bit steeper before the correction, and then it sort of shifts here. And I didn't really go into detail about this, but there's some work by Michael Lynch, and he suggests that we don't really need to worry about correcting for these things because the coalescent times are much less than divergence times. And I have some figures in the paper where I show that, yes, I mean, that's true, but what's determining the coalescent times, like effective population size, are variables that have phylogenetic inertia, right? And we can actually see, so there's this thing they do in the phylogenetics literature where they compute basically the standardized contrast, and then they look at how these contrasts change across time, basically depending on the node and the phylogeny. What's fascinating is that if you do this for diversity as a trait, you see that some, like I think 600 million years ago, there are some nodes where diversity as a trait suddenly changes in its evolution. And these correspond actually to the split between chordates and tunicates, right? And we know that C-squarts have unusually high diversity. So I think that just in general, there's some fascinating stuff to be done at the intersection of population genetics and phylogenetics. All right, so let me keep reading here. If it isn't link selection that explains Lewinton's paradox, what's your best bet? What does? So as I mentioned, I'm in the camp that these really high census size species, which also have large ranges, have really, since the last glaciation, have expanded the ranges, and there's probably some like recurrent bottleneck and founder effects. But I think an underexplored area is this idea that there may be some interaction between macroevolutionary diversification processes and demographic processes. And the idea here that I propose in the discussion of the paper is that if we imagine that there's variability in the diversification rate, where really large census-sized species are diversifying faster than they can sort of hit mutation drift equilibrium, then there would be perpetually sort of reduced effective population sizes at this tail. And I think that this is something which, to my knowledge, hasn't been suggested in the literature, but I think that could be at least playing somewhat of a role in this. I mean, you look at things like Drosophila, and there's just so many little species that have sort of broken off since during radiation, and we're only considering diversity within these species. So there's some interplay of population genetics and macroevolutionary processes there. Let's see. And does the inclusion of density affect the pattern very much? Much shallower than we see in mammals and birds. I don't know what the inclusion of density means. And population density? Is that what? Yeah. So this is a good question. This is, I think, to really answer Lewinton's paradox, which I always frame as this, again, really broad across metazoan process. But there's some papers that look at it at within a clade or something, and maybe that's what you're getting at here. So I think that's going to be part of the problem. So you can imagine that if you have a mainland species and an island species, that we know that the census sizes of the island species are going to be reduced, and we can sort of date the sort of split between these two things, and you can sort of assess like to what extent reduction in census size is affecting diversity. And I think this is one of these exciting areas where it's going to take macroevolution, macroecology, and population genetics to really suss out how these processes play out, because that recent split and change in effective population size, I think, would lead to a steeper sort of change between census size and population, and pairwise diversity, because there's not this overlay of all of these other processes. So we'll see a stronger sort of reduction in diversity due to that kind of thing. So Derek has a question here. Would haplotype structure in the initial population at the beginning of the evolve and resequence experiments be a nuisance for your covariance approach in estimating the contribution? So yes, this is, I don't know if I would phrase it as a nuisance, but so we explored this with some simulations, and we looked at how the size of sort of the ancestral population affected things. And with the idea that if it's smaller, drift can lead to linkage disequilibrium at a faster rate. And our simulations confirm that, essentially, that what'll happen is that, if you have a very small founding population, you end up with really large haplotypes, and this can actually inflate sort of the convergence correlation when these later replicate populations experience selection. So that was a little bit of a whirlwind question thing. I was trying to keep up with them. Are there any others?
Yes, I'm just gonna do like a few announcements, like just to give people the chance like to make their questions as well. We have a few minutes left. We have 10 minutes left. So anyways, I'd like to tell you that if you'd like me to invite people to participate on CGDG, if there's a paper that you want, you can write me on my email and I'll write them myself. I was able to get Vince. So like if there's any speaker that you'd like me to invite, like I'm pleased to do so. So like just send me an email. Um, so yes, I was just wondering like did you use a particular software for the simulations that you carried, or like did you develop the software yourself?
Yeah, that's a good question. So in our 2019 paper, the genetics paper, I wrote all of those simulations by hand using R. And so what I would do is burn in the population using haplotypes that I simulated from MS Prime, and then I would sort of randomly choose alleles in those haplotypes that would affect the trait. Then I evolved them forward in time for 20 generations or something in R. And that doesn't scale well at all to large population sizes. R is slow, as I'm sure you all know, for this kind of thing. But it allowed me to like really look at what was going on and explore things interactively. So for the PNAS paper, what I was doing there was using SLIM from Ben Holler and Philip Messer's lab, which is just a fantastic piece of software. For the most part, I've shifted everything over to SLIM, and it just sort of keeps getting better. So I'll be using it for some time. But that allowed me to do a lot more realistic simulations. And it's also quite nice because you can save the population state at a particular generation. So I was able to basically burn in ancestral populations, save that, and then prime them into different replicate population splits and save time that way.
Right. Well, thank you very much, Vince. I don't believe we have more questions here today. Guess I'll see you all in our next meeting. I'll let you know by email invitations. So yes, thank you very much for attending, you all, and thank you, Vince, again.
Thank you.